Hello, my name is Caroline Schlehuber. I am a senior at Visitation School, and today I will be explaining my research project exploring antibacterial properties of plants and fungi used in indigenous medicine. I encourage you to pause this video at any time along the way so you'll be able to read a more in-depth version of my research. To begin, the problem I wanted to tackle through my research was the serious lack of effective antibiotics and antibacterial medicines in our world today. And the angle I wanted to take with this issue was researching the medicinal practices that have already been used for hundreds of years in indigenous cultures. To do this, I chose two plant species and one species of fungi that were historically used in indigenous medicine, and those were Canada goldenrod and stinging nettle, and then the mushroom lion's mane mushroom. And so all three of these plants and fungi were used by a variety of tribes across America to treat everything from burns to digestion to arthritis, pregnancy, wounds, you name it, there was a use for it. And I will talk about how I studied these in my methods. So my specific goal was to find some kind of antibacterial activity or resistance through inhibition assays. My general goal was to add to the conversation that there is a lot of merit to indigenous medicine, and it needs to be held with more value in Western medicine. I will reach this goal through some controls, like using the processes found in other research articles that did similar research, and by having controls in my testing. And I will know that I found success by proof of bacterial resistance in my specimens. To begin, I first gathered samples from the field. It was really easy to find the stinging nettle and goldenrod because they're really just viewed as weeds in kind of modern culture. And I was really just lucky to find the lion's mane mushroom. So that was really exciting to me. I stored the samples, sterilized them, I dehydrated the fungi, and then I next placed the plant samples on cornmeal agar and I grew endophytic fungi, or fungi that were living within the plant, onto the plates. I then took plugs of this endophytic fungi and let them keep growing in a more controlled environment. I created an extract out of the lion's mane mushroom, or Herinaceum erinaceae, for testing, and then I performed inhibition assays on both the plants, or the endophytic fungi, and the extract of the fungi. So for the endophytic fungi grown from the plants, I placed plugs of that fungi onto plates and let it grow against bacteria and against other strains of fungi. And then for the Herinaceum erinaceae extract, I placed it on paper discs, and I also injected it into the agar, and I examined the differences between those two methods. And finally, I measured the rings of inhibition and came to some conclusions, which we'll discuss later. So these plates show how I was able to grow endophytic fungi out of the plants. So these cloudy areas that are surrounding the plants, on the top row of plates are hyphal threads, and I took samples of these hyphal threads and replated them onto potato dextrose agar, and I grew these species of fungi which were living within the plants. So these plates are proof of antifungal and antibacterial resistance. So first, this plate on the far left shows sample A, which was a species of endophytic fungi that showed strong potential for antifungal activity, which was really something I wasn't expecting to find. The middle plates, the 6-well plate, was the first evidence of antibacterial activity that I found for the Herinaceum erinaceae extract. And so the plates circled in red are the extract against 2 strains of bacteria. And so you can clearly see the rings of inhibition that were created by this extract. And then it's the same thing on the plate on the far right. Again, this was just a repeated trial of my testing. And I was testing the extract against Enterobacter aerogenes and Staph epidermidis, which are 2 strains of bacteria, 1 gram positive and 1 gram negative. So this is a data table measuring the rings of inhibition that were created by the Herinaceum erinaceae, or lion's mane mushroom extract, against both strains of bacteria that I used. So one trend that I found that was really interesting was that as time went on, the rings of inhibition grew smaller. So that's something I'll talk about in my conclusions. But as you can see, the rings of inhibition for all of my testing were around 1.5 centimeters, which is pretty significant in terms of inhibition rings. So I drew a number of conclusions, beginning with the sample A that I showed on one of the results slides and how it has potential for antifungal properties. Next, like I mentioned, I found that the diameter of inhibition rings for the lion mane's mushroom extract, it is directly correlated to the age of the mushroom extract. I also found that Herinaceum erinaceae inhibited the growth of Enterobacter aerogenes more than the growth of Staph epidermidis. For future testing, I would change how long it took me to do the experiment. I would kind of do it all at once instead, and I would also keep the extract in the refrigerator because during my experiment I left it sealed in parafilm, but at room temperature, and I think this was kind of a cause for those inhibition rings growing smaller in size over time. And a future question I have for my research is how we can calculate the appropriate dosage of mushrooms for medicinal use. I would like to thank my adult sponsor, Mrs. Peel, and my school for helping me make this project possible. And here are my sources. Thank you so much for watching. I hope you're able to learn something today, and thank you for your time.